Thanks for joining us today for our Plant Phenomics Fridays summer seminar series that we put on for the month of August. If you listen really carefully, maybe you can hear the carillon at Iowa State, because that's what's going on right now is the noon hour carillon uh, serenade that they do most days while school's in session. What we're doing today is to have two short seminars, one from Adina Howe, one from Zihao Zheng. And the way that we'll manage that is as we have the last three, where the first thing that we do is one seminar, and if there's time in that first 20 minutes for a question or two, we'll do a question or two, but if not, we'll roll directly into the next one. And once that second seminar is finished, if there's time for a question or two, we'll do that. And then there should be time at the end for 10 to 15 minutes of discussion that maybe it draws on both seminars, maybe you're interested in both speakers' perspectives. You can think of it that way. The seminar series itself is one that we've organized before, Plant Phenomics Fridays. It's in person at Iowa State, but also available to all of you online. And if I scroll down, you can see not only who our speakers are today, you see the links at this website, phenomics.iastate.edu, to the recordings from the last few seminars. And you can also see who has funded our seminar series. USDA NIFA funds the HIPS project, and USDA and the NSF together are funding the IRA, AI Institute for Resilient Agriculture. That's where we're getting support to organize this today. Our first speaker is Adina Howe. Adina is an associate professor in the Department of Agricultural and Biological Engineering at Iowa State. She got a BS from Purdue in mechanical engineering and MS in environmental engineering, also from Purdue. Then at the University of Iowa, got her PhD in environmental engineering in 2009. Adina's seminar title today is Plant-microbe Interactions in Bioenergy Production. And with that, I will turn it over to Adina.
Thank you for the introduction. I'm going to be talking today about a collaboration that's quite extensive, and it's a collaboration between two Department of Energy biorenewable centers, including CABI, which is headquartered at the University of Illinois, but has a ton of different collaborations across the U.S., and also the Great Lakes Bioenergy Research Center, which is Wisconsin and Michigan State mainly, and again, a lot of different collaborators. But mainly, let me highlight that this is a collaborative effort with myself and my team and Ashley Shade's lab at Michigan State. And so we've worked together on this slide deck, but she gave me, I think, the best slides over my slides. So a lot of the slides are hers, so I want to acknowledge that to start with. And our team here includes Shane Dooley, Fan Yang, and Nate's Stopsisnik, and Kira Grady. All right. Why talk about plant-microbe interactions? Maybe you've heard over the decades of research to talk about pathogens, the pathogenic microbes that are influence plants. Increasingly, we're becoming more knowledgeable that there are also benefits that microbes can provide, including drought tolerance, stress tolerance generally, nutrient cycling, things like nitrogen fixation, and also disease suppression, right? So we know some things based on recent research about plant soil microbes. They're responsive to the season and changes in the environment. Where you study the microbiome matters, whether it's in the roots, in the soils, bulk soils around the plants, the stems, the leaves. Management practices also matter, whether you've had fertilization in the past, whether the soil's been tilled or not. Exposure and space influence microbes. And we also have to consider the very dynamic nature in this system. Microbes move back and forth from the soil to the plant and back again. And that creates all sorts of complexities, and especially understanding plant-microbe interactions in the field. Okay, so why talk about biofuel feedstocks? Plant-microbe interactions can help plants be productive. They're a part of what we're thinking will help produce sustainable biofuel feedstocks. Biofuel feedstocks, in turn, are part of a portfolio to mitigate climate change, right? And this slide really shows nicely, I think, what that solution might look like. Biofuel feedstocks are going to be providing the biofuels themselves, In addition, some of these feedstocks, especially perennial feedstocks, can help carbon capture and storage. And so these are parts of our plans and the Department of Energy's plans to provide sustainable solutions for energy. So the feedstocks that I'll be talking about today are two perennial grasses that you may or may not know of, but switchgrass and miscanthus. And what we want to do is learn to grow these on marginal lands, lands where you might not typically grow food crops, might not have a bunch of nutrients, and might not typically have agricultural productivity. We want to grow them with minimal inputs, especially like nitrogen, which can contribute to water quality issues. And we want to grow them in places that might be harsh, where we might not have a lot of water. And so how are we going to do that? And one of the thoughts is we can start thinking of managing the microbes that interact with these plants as a resource of that management. So today I'd specifically like to talk about the phyllosphere compartment on the plants. The phyllosphere is anything touching the leaves of a plant. So why the phyllosphere? I think it's easy to think of biomass production and leaves, right? They're directly connected. Second, the phyllosphere is actually the largest environmental surface of microbial habitation on the planet. It makes up 100 million kilometers squared of surface globally. And so it's about a 1/5 of the Earth's microbial habitats are made up of leaves and the microbes on these leaves. So they're incredibly large habitat for microbes, but also an incredibly understudied habitat for microbes. Microbes that live on the leaves can confer benefits for the plants. In other plants, not necessarily the perennials we're studying, but we know they increase stress tolerance, promote plant growth and reproduction, and protect from pathogens. Extending these impacts more broadly, this means these benefits can convert to moderating methanol emissions, contributing to global nitrogen fixation, and looking at also minimizing other greenhouse gas feedbacks. So the question or the challenge that we were presented with is how do we manage this plant microbiome in the phyllosphere to promote biofuel production, specifically crop vigor and resilience? And specifically how we interpret that is when we look at microbes on these plants, when we can identify them, which we're really good at identifying the microbes on plants, how do we distinguish microbes that provide benefits to the plants that aren't just commensals? They're not just living there and eating whatever's there on the plant just for their lifestyles, but actually providing direct benefits to the plant, and also transients. And also, how do we understand how these bacteria are changing over time or under management conditions? So today I'll tell you a little bit about how we have done that for miscanthus and switchgrass. Okay, so how do you study field microplant interactions? It's easy enough to bring a crop into the lab, maybe, arguably. Actually, I would argue it's kind of hard because you can't mimic what it sees in the field. Often you'll see a small subset, and that subset might not even be representative of what's seen in the field. So we worked in the field site of the Kellogg Biological Station, which has replicated miscanthus and switchgrass, along with other bioenergy crops, and also corn and fallow. But we are looking here at, I wanted to show you kind of the plots that we're looking at. Switchgrass and miscanthus are in the orange and dark kind of copper red. And if you're colorblind, just know that they're well replicated in a random plot design across this experimental station. So we go out to these different plots and the different replicated plots that they have at this system, and we sample the leaves and soils of miscanthus and switchgrass over time, from April to November. And this captures the pre-emergence and senescence of these perennials. And we sample also under fertilized and unfertilized conditions. All right, so once we get the leaves and the soils, there's a number of measurements we take. One is looking at the plant side of things, looking at the leaf data and soil chemistry. From the microbial side, we wash the leaves off, try to get the microbes off, and we extract the DNA, which then gets used for metagenomic sequencing, and I'll talk a little bit more about that, and also phylotyping, and we also extract the RNA, which looks at the transcriptome. We also do a suite of different characterizations of microbes. Today I'm going to be focusing mainly, on the DNA and RNA analysis of what we saw on the phyllosphere of the miscanthus and switchgrass. So why look at the DNA and RNA? And I'm going to make an analogy here that maybe will put things in context in case this isn't something you're very familiar with. If you think about the DNA of the microbes, that might be analogous to a recipe book. Everything the bacteria on that leaf can do, potentially do. And so that's important because then you know what that bacteria can actually potentially do. But then you have to overlay that of what your meals you're actually cooking. And that's what the RNA is going to tell you. So that's going to tell you what those bacteria are actively doing at different times. And both of these pieces are important to understanding what those bacteria can do and are actively doing and thinking about how that links to the benefits of the plant. So the DNA is the potential, or again, the recipe book, and the RNA is the meal that's cooking and it's actively growing. All right. I would go into lots of results here, but for the brevity of time and for the sake of conversation, let me tell you some of the take-homes from first our observations from the DNA. And from the DNA, you can do one thing really easily, which is phylotyping. Who is there? What's the microbial membership? What's the community look like? And how is that changing in response to time? From that, I can tell you that there's a core taxa that's present in miscanthus and switchgrass on the leaves that are persistent across the seasons. But they exhibit seasonality. Sometimes they're enriched late in the season, sometimes early in the season. These taxa are observed to be pretty abundant in the microbiome, these core. Remember, we're trying to differentiate them from commensals or transients. And they're consistently detected across both perennials. They're not just miscanthus or switchgrass related. They're in both. So that's interesting. That was our first rationale to push on to looking at other data sets, which are more complicated and more expensive. And these other data sets, then our focus is what are these bacteria doing? Now, we now know they are there, but what functions are they providing, and are they providing benefits? How are they engaging with the plants? Are they even active or alive? Are they just sitting there dormant, and we know that their recipe is there, but nobody's actually cooking a meal, or are they actually growing? So these questions, while they seem very simple, are still unanswered in phylosphere research. Like, are the bacteria on the leaves even alive? We know they're there, but are they even alive? So we looked at the RNA and DNA, and I give you this data slide mainly because they will frame some of our results. I wanted to show some results in this brief conversation of the DNA, which are metagenomes from miscanthus and switchgrass over 2 years, and also the RNA from just switchgrass from 2016 and 2017. Why do we have this number of samples? The rationale is cost. These libraries, these sequencing results cost probably $500 per sample on the low end, right? And the RNA probably even up to $1,000 per sample, I would say. Why doesn't everybody do this? Why? One of the things that I won't be able to really help you appreciate is the complexity of extracting microbial DNA mixed with plant DNA is outstanding. And it is a barrier for micro-plant interactions right now. And I wanted to say that because these are actually the first results that I know of from agricultural soils with RNA in the field looking at microbes and plants interacting together. And so that's what we're really pushing this is being reviewed right now as the novelty of this study. Okay, so what do we see over time in the perennial grasses? On the left, we have miscanthus. On the right, we have switchgrass. And on the x-axis, we have the season, from early season, from pre-emergence to senescence on the late. And on each row, you can think of it as a microbial member or a recipe, if you will. What recipes do we observe? And we see an enrichment over the seasons. You can see that there are multiple microbes present that are consistent among both switchgrass and miscanthus. You can see that they're present in both plants, and you can see that over the season they have different dynamics. But mainly you see this pattern that they're enriched at the end of a season, right? We see this in the DNA, and so we were wondering, are they alive, and are these patterns also true when we look at what meals are cooking? And indeed, we see that is true in the switchgrass for both seasons, that we see this enrichment of this activity throughout the season. And so we see these seasonal patterns, and we can actually go into these genomes and figure out what functions they confer, and that's what we did next. And we had two big questions that we're trying to figure out what functions. So how do you figure out, when you see these functions, how do you decide what's important? So we had kind of two criteria. One is, what functions are shared among this membership that we're seeing as core and active on the phylosphere? If it's shared, that might give us some insight to something that's important to the plant, right? That's one piece of evidence. If it's shared between membership, those functions might be providing a benefit or some reason that they would exist on the plant. It's weird to grow on the leaf, right? It's a pretty nutrient depleted area on the plant. So I would say it's a harsh, extreme environment. But you can't just look at what's shared, because we all know that this is also a niche environment and there might be specialization within the membership. So we're looking at both pieces. To summarize what we did see, though, we see enrichment of a few interesting things in the transcripts. We see enrichment of pyruvate metabolism, which is actually a regulatory switch for controlling both carbon and nitrogen in response to, in model systems, they've seen that pyruvate metabolism can control nitrogen depletion. So you might see increased pyruvate metabolism in response to low nitrogen conditions. We also see isoprene biosynthesis. This is an interesting signaling molecule to tell that chemicals get released from the plants, and potentially the microbes are responding to those chemicals. And we also see tree hallose biosynthesis, which is an indication that we're seeing stress response functions in these microbes that we're seeing on the phylosphere. All right, to sum up, the phylosphere microbiomes this is the first time we've seen evidence that they're actively growing, and they appear to be seasonally active. They're also broadly present in perennials and across geographic boundaries. We don't just see them in the Michigan site, we see them at our Iowa site and also at our Wisconsin site. So these microbes may be resources to think about, maybe enriching in marginal land conditions or inoculating in marginal lands conditions to confer some benefits to these plants. The next thing is more mechanistic studies looking at these specific functions, and we're really interested in following up with the isoprene biosynthesis and the stress response functional pathways to target in these plants. So we have a suite of isolates also from the phylosphere that we can then now put with the plants and do more mechanistic studies. So just going back to kind of the bigger picture, how are we going to grow these plants on marginal? So one of the things we like to think about is, do these functions pair with our goals to growing productive biofuel crops? So metabolism, signaling, stress response, those are all functions I think of needing to leverage as a resource if we think about how we need to grow plants on marginal lands where nutrients are depleted, with minimum nutrients, and in stressful conditions like drought. With that, I'll just acknowledge our teams and also our funders at the DOE. I think I've got time for a couple of questions.
Sample to get the data. What kind of irrigation were you guys doing? Were they regularly irrigated? Was it just rainfall? Because from what I know of phylosphere, water is a really big factor.
Yeah.
And it can also be a big source of introducing new bacteria,
Right.
Such as rainfall.
These specific crops and also the parallel study crops here at Iowa State are not irrigated because, well, I think we're trying to study them and growing them in marginal lands. Our plan is not to add more water to them at this point. The other part of the piece of the puzzle is they're also tile drained, so that there's other studies going on to try to complete the water balance of these crops. So that's also another piece of study. You might. So we have 2 years of study for a reason, because we have different seasonal dynamics in both those years too, and we saw similar seasonal trends. So I suspect that the scope of the study wasn't there to study the impacts of rainfall, though I'm sure it has an effect. At the broad pieces right now where we see the biggest variation, it seems to be driven by season more so than even plant type, which gives us some thought of, like, I think that's good, because if it was microbes specific to miscanthus or switchgrass and specific to a site, it's going to be really difficult to implement them as a resource broadly, right? So I think it's good to understand their broader context. And the results seem to suggest that that gives us some opportunity to think of them as a resource.
Do you think microbes are there as a phenotype for the plant?
Yes, I do. And I think that we need to be increasingly thinking of that. So, one of the things we haven't done, and I think people are very interested in the centers, is matching host plant phenotype with microbe phenotype. One of the problems with that is we don't even know which microbes to chase right now. And so I think this sort of study in the field that is a field-driven, derived, these are targets of interest, can give us insight into what to chase. Because we can't have 10 different hosts and a gabillion different microbes. That's going to take so much effort for discovery. So this is kind of a discovery-based targeting, setting up the next experiment. But yeah, we do think of it as a phenotype.
It's really interesting. It seems like a different way of thinking of association.
Mm-hmm.
Are there questions on your screen here? Anybody got any questions before we turn it over to Zihao? Oh, we got another question in the room.
Sorry. So, just out of curiosity, when you did the abundance, was it relative abundance, or did you actually do an absolute abundance by like spiking it with something, or?
Yeah, so there is no spike in our omic libraries, though. Unlike phylotyping, where you'd have a percent of the total observed community, we can get numbers per cell, estimated per cell, because we have both. The sequencing allows us to capture both the numbers of regulatory genes or housekeeping genes and functional genes. So it's a normalized abundance, but not a relative abundance. There was
another question. One more, and then we need to move.
Yeah, while
next
you're walking, Carolyn,
speaker.
there was a question online. Did we capture the fungal transcriptome and the metatranscriptome data? We did not, and the reason why is if you're going to do fungal transcriptome, what you should do is a specific library prep to capture the poly-A tails to really enrich for the fungal, and we didn't. That was not the intent of this library preparation. We really wanted to look at the bacterial, but I think that's a great idea. But I think it would have taken another sort of library preparation, because you can take advantage of the eukaryotic properties of fungal to enrich for them, and then deplete. Actually, fungal is easier in ag metatranscriptomes for that reason, because you can enrich for it.
Yeah, is there any commonality between soil microbes and philosphere microbes? Because you had done soil core microbiome as well as the philosphere microbiome.
That was actually a part of the study, was to figure out where the origin story of the phylosphere comes from, and they come from the soil. That's the data suggests they come from the soil.
The question is, are they going back to the soil?
Yeah. Okay. So the origin story is the question is on the origin story, and then maybe the return story. I don't know if that was looked at. That's a good question. I would not be I think it's a little harder to chase, because you'd have to really have something to trace them. I don't think we could answer that now that I'm thinking about it, because you can see them, they're not in the leaves going into the leaves, but then they're already in the soil, so how do you distinguish them going back? And you'd have to have some sort of tracer element in there. Yeah. That's an interesting question.
The question is about whether the microbes are in the air as well and whether those are captured.
Thank you. Thank you for repeating the question. That's a great question and has a lot of impacts on pathogen distribution, maybe not in this study. It's outside the scope of maybe the bioenergy study, but we have a lot of questions on that and things when we think about pathogen emergence and persistence. But I think that it takes a whole nother team to figure out how to capture the air, and it's pretty dilute in the air compared to enrichment in the soil, so you'd have to be very clever, I think, a little outside my expertise.
I really love how questions are slow to start and then everyone starts to have them. And my hope is that as we go through our next seminar, that some of the discussions and questions will be brewing in your head so that we have a rich discussion following maybe a question or two for Zihao. Zihao is a PhD candidate in the Schnabel Lab at Iowa State University. His research focuses on utilizing large-scale genomic and transcriptomic data for candidate gene discovery in maize by way of genome-wide association studies and trait predictions using machine learning models. He also works closely with engineers and computer scientists to develop high-throughput phenotyping pipelines for novel traits, novel trait extraction from images. You see Zihao's title here, Prediction of Maize Root Lodging, using architectural traits of root systems.
Yeah, thanks for having me. My name is Zihao Zheng. I'm a graduate student from Dr. Pat Schnabel's lab here at Iowa State. And today I'm going to talk about our latest work on root lodging. So lodging is among one of those important factors that have significantly negative impacts on crop yield. So the definition of lodging is plants' displacement from its original vertical positions after some inducing environmental events, such as heavy rainfall or strong wind. So worldwide, the annual yield losses that attributed to lodging are about 20% in all major crops, including maize, rice, and wheat, etc. So depending on the position of the occurrence, lodging can be further categorized into stalk lodging, as shown on the top panel here. So the stalk just snapped. And also, if it happens around the root areas, it's called root lodging. So stalk lodging or stalk breakage can lead to partial or completely yield loss if it happens at the later stages of the field season. This trait, stalk lodging, the mechanisms are relatively well studied in terms of both physiological mechanisms and also genetic basis. So, for example, in this stub et al. Study, they modeled the stalk strands node by node. And there are also other studies linking these model parameters to actual anatomical structures in stalks and also the chemical components of stalks. And in terms of genetic basis, there are a lot of genome-wide association studies or QTL mapping that researched the genetic architecture of this trait. And also through mutagenesis, there are a few genes that have been cloned about maize stalk lodging. For example, this recently published one, they cloned a gene, maize gene called STIF1. So the overexpression of this gene can lead to stalk lodging in their experiment. So for root lodging, things are a little bit tricky here. So the definition of root lodging is the failure of the root system to anchor the plant to its original uprightness. So the bottom figure shown here says that root lodging is prevalent, although it varies from years to years. It's kind of prevalent in the US, especially in the Midwest. So there are a substantial amount of work on modeling root lodging, for example, via satellite images data or UAV, etc. But there are very few studies that looked into the genetic basis of root lodging. So far, we know that there's one QTL in maize called root ABA1 that have been identified and validated. And we also know that there's one gene called maize agro8. The overexpression of this gene can lead to root lodging. So the reason, some reasons why root lodging is less researched than stalk lodgings are the trait is kind of plastic. So there's plasticity of root lodging. So when the root lodging happened, some of the genotypes are able to partially recover from root lodging through a phenomenon called goose snacking. So that is demonstrated on the top figure here. And also, so this makes the scoring of the trait root lodging much harder if you're using satellite data or UAV. And also the expression of this phenotype, root lodging, largely depends on the occurrence of environmental effects like storm. So this would give the researchers much less control in terms of experimental design. So it's very hard to have, say, an ideal set of materials that are affected by the same storm or having the same set of materials over many experiments to see how the same genotype vary from environment to environment, from storm to storm, and score the root lodging for these materials. And last, it is not trivial to phenotype the root system since it's underground. If you want to study them, you need to dig them out and phenotype somehow. So that's why to date, many of the works have been focusing on assessing the impact of aerial roots or airborne roots, like brace root here, impact on root lodging. So on the August 10th of 2020, last year, a complex of strong wind and heavy rain, known as the derecho, swept through the Midwest, all the way from East Nebraska to Indiana, and left a path of destruction on crop fields in these states. So in Iowa, it affected more than 8 million acres of corns and over 5 million acres of soybeans, and caused a huge damage to farmers. And the epicenter of this derecho in Iowa are actually Story County and Boone County. For those of you who are outside of Iowa State, Iowa State is located in Story County. And in that year, all of our fields of experiments were in these two counties. So we took this opportunity to study root lodging. So we made a protractor using a cardboard and used this very easy system to score the root lodging for a diversity panel from 3 existing experiments. So the scale works like this. So if it's 1, it means that it's totally upright. So it's not affected by the storm or the derecho. And the larger the number in the scale, the more severe the lodging. So 5 stands for completely lodging in our case. So as I just said, we scored the plants from a diversity panel consists of 369 genotypes. It's also referred as the SAM panel. So we scored more than 2,100 plots. So these are 2-row plots or 4-row plots that mimics the agronomical conditions in the real world. And also for over 30,000 individual plants. And for this panel, we have over 1.2 million SNP markers, and also gene expression data from 8 tissues. So we can do genome-wide association study to dissect the genetic architecture of root lodging. So with this panel, we did GWAS on root lodging for each experiment we scored separately. So in total, we were able to identify 25 unique root lodging associated SNPs using the cutoff of the false discovery rate of less than 0.05. So this is an example of a Manhattan plot of root lodging at one of our location in Story County from 2-row plot low density experiments. So out of these 25 trait-associated SNPs, there were 24 candidate genes we were able to identify. And 9 out of these 24, which counts for 38% of the genes, are highly expressed in root tissues based on our RNA-seq data set. So this led us to the hypothesis that maybe we're able to use the root architectural traits to predict for root lodging. So in the summer of 2017, we created a pipeline called Cream Coffee, which stands for core root excavation using compressed air, and a software called Coffee, core root feature extraction. So we were using this system where we use this highly compressed airs to blow the dirt away around the roots and excavate them out to phenotype them. So with the same panel we used in 2020, we excavated more than 3,000 plants from this panel, and we took photo. We have more than 6,000 of 2D images of the root system. So with this 2D images as input, our collaborator, Dr. Zaki Jubri, trained a unsupervised deep learning framework called autoencoder. So it has two parts, the encoder and the decoder. So we use the input of the encoder were the images we had for the root. And the encoder took the input and compressed to it to an extent that only the key features of the images were kept. And with these compressed key features, it went through the process of decoder, where we did reconstruct of the input, the input images. And by comparing these, by tweaking the similarity of the autoencoder output versus the input, we ended up having 1,032 root latent features that capture the structural traits of the root system architecture of maize. So with these latent features, 1,032 root latent features as inputs, we used a random forest model to predict the root lodging for this panel. So we did a 20/80 split of training set versus validation set. And the mean correlation coefficient was 0.61. So currently, we're still in the process of optimizing the autoencoder for latent feature extraction, and also parameter fine-tuning for the random forest models. Yeah, but these are our current status of the project. So in summary, we investigated root lodging for a maize diversity panel consists of 369 genotypes after the 2020 deratio. And we were able to identify 25 trait-associated SNPs and 24 candidate genes through genome-wide association studies. And a large portion of them were highly expressed in root tissues. And we also demonstrated that we are able to predict the root lodging using the architectural traits of root system through the latent features we extracted from the images. And that is all I have for today. And with the rest of time, I would like to thank our lab members and our collaborators at Iowa State and outside Iowa State, and also take some questions.
Thank you, Zihao. Zihao, and do we have any that are online? I see one in the room.
Yeah, go somewhere else first. Yeah, that's good. Students should go first.
A nice presentation. So when you are washing the roots, like using the compressed air, did you damage the root while phenotyping, or how that works? Like you are applying high pressure to the root, then
Right.
you are imaging the root system. Do you have any trade-off in that?
Yes, there are definitely trade-off on that end. So we excavated the root around the flowering time. So the root was already heavily lignified. So we did lose some fine structures of the root, but we kept the overall structure relatively intact.
There's another question in the room.
I do have a question.
Yeah.
When you consider the roots, did you also include the braced roots or not?
Yes, so we took photos of the so-called core root system or crown roots of the plants. So in the photo, it actually includes the brace root from the base node of the plants. But if the brace roots were on the nodes more above, then they're not included.
Thanks for the talk. I thought that was very interesting, and I think it's very clever to study this after the derecho. I had a question on the derecho is a pretty, hopefully a rare event, and the applications of these results to broader, more agricultural settings, and how you guys are using this to interpret that. But I definitely, I don't want to diminish how I think it was very unique to use the machine, because you need that gradient of root lodging to happen to create that model, but then how broadly applicable is this more conventional settings?
Right, thank you. So actually, so this is just part of our project. So we also had this root lodging data from a data set called the G2F initiative, the Genomes to Field initiative. So that's more than 20 states in the US participated. So they planted the same set of maize hybrids over multiple years, and they scored for root lodging, stalk lodging, yield for many years. So that gave us the opportunity to see how root lodging vary from years to year or environments to environments. So we also have this same set of latent features for the a panel of hybrids that is part of the G2F initiative here. So that's also something we're trying to try to predict the root lodging. So that is not from such an extreme case like the ratio, and I think that's more agronomical relevant.
Do the features that you guys are finding to the derecho overlap with the features you guys are finding in the genomes to field models?
I'm sorry, can you say that again?
Are you finding the same? Is the models similar? Are they different? Where you would think the features that are predicting root lodging are the same for the derecho crops versus the genome to field crops?
You mean the candidate genes underlying these two sets of panels, or the root architectural features that has the highest?
The candidate gene. That's fine.
Oh yeah. So for the G2F hybrids, we only have about 100 lines. So that is not large enough for genetic mapping. So yeah, so we don't know the candidate genes underlying those materials.
Do you think that the soil moisture and the soil type could have an effect on your study?
Definitely. So we know that there are several factors can affect root lodging, including soil moisture and planting density, etc. So for the G2F hybrids, we are calculating the reaction norm, or how the traits varies across environments for specific environmental factors, and soil moisture is one of those factors we are studying. So yes.
So I have a question, and it goes back to the question, I suppose, that I asked earlier of Adina about whether she considered what the microbiome is, who's there, to be a phenotype or not. Mm-hmm. What do you do when you have a trait that is multidimensional and you're trying to do association genetics? So, for example, if I think of something like dwarfism as a phenotype, the plant's short, its internodes are short, its leaves are broad, the leaves are long. There are all of these different pieces of that phenotype, right? Yeah. And if I think of what the microbes are that are present as being a multidimensional phenotype, what are the ways that people analyze that kind of a problem? I don't ask with an answer in mind. I ask because I don't have an answer.
Yeah, I don't have an answer either, unfortunately. But I think what people do are either they do some kind of reduction of the dimension, like the PCA or something, to capture the variance of these high dimensional traits, or you can go through them one by one to loop through or prioritize some of them somehow. But yeah, this is a very interesting question.
I was curious. This is Klaus Goller from Corteva. I'm curious. I was curious. What was the heritability of the phenotypic data that you had for whatever you were using for root lodging for this trait?
Okay, so I think as I mentioned in one of the slides, we scored root lodging from 3 experiments, and the heritability varies from experiment to experiment. So the lower end of that is around 0.5. The higher end of that is around 0.78, I think.
Well, that's very high. I mean, I would say, for these kinds of, you were lucky, I would say, for root lodging studies, that's very high. Thank you.
Yeah, thank you.
Any other questions or questions that are more sort of general for discussion? We have a few more minutes that both Adina and Zihao are available.
Oh, I think there's a question
Go
in the
ahead.
chat. Yeah. Do you think the end of season plant height is useful for root lodging? Is different of plant height at the end of season and immediately after to reach you a useful metric? And this question is from Kaushik. Yes, it is definitely a very useful approximated traits for root lodging. As a matter of fact, in this paper I cited here, so they were using UAVs to assess the impact of storm on root lodging, and they were using the end of season plant height versus the in season plant height they had as a index for root lodging. But the problem of that, one of the problems of that is, as I mentioned in this slide, on this slide, so some of the genotypes are able to partially recover from the lodging. So that would make the case more complicated. So by using this quick and dirty way to score the root lodging, and these photos were taken the day after the ratio, so that's August the 11th in 2020. So we were able to capture the variation of root lodging to avoid this goosenecking to affect our scoring of the phenotype.
There will also be root rot diseases might affect the lodging.
Yes.
So how do you put that into consideration, just to be truly through the wind rather than root disease or stalk disease like anthracnose or fusarium?
Yeah, so thank you. This is a really good question. Because back in the 1960s, there were hypotheses about the trade-offs or the dilemmas between the source and sink and the insect effect on root and lodging. So if the root has like root worms, that would definitely increase the chances of the plants for root lodging. But for our case here, at least in 2020, root worm was not a huge problem here in Ames. It might be the case for some of the environments we had for the G2F initiatives data. I'm actually not sure on top of my head if those were documented or not, but if we see some like weird distribution of this phenotype, like it's super severe at one location, and the weather data tells us there were no strong wind or rainfall, then there might be rootworms or other insects that affected the root lodging there.
Thank you.
Thank you. And there's another question in the chat from Dr. Xingwei. He said, after the storm, he noticed some stalk lodging, or you noticed more stalk lodging or more root lodging. Is there any possible relationships between stalk lodging and root lodging? Yeah, thanks for the question. We actually saw both root lodging and stalk lodging, and the percentage of stalk lodging we had in this experiment is about 20% of the genotype. And root lodging, it's much more prevalent than stalk lodging in our case here. So for the question, is there any possible relationship between stalk lodging and root lodging? So root lodging and stalk lodging are likely due to two very different mechanisms. I'm thinking about genetics right now, but I think it's totally possible that these two traits are somehow, there are some interplays between these two traits. So I don't know the answer, but I think my hypothesis would be yes. Thank you.
We have time for one last question if there is one more. Sometimes some questions spur new ideas and new questions. So if you've had new thoughts that you want to ask another question, please go right ahead. I see one now.
Did you observe genotypes having large root lodging but higher recovery? Yeah, I think this is a very interesting question. I don't know, because we didn't pay attention to the degree of recovery of the plants. So I think this question is more about the plasticity of some certain genotypes, the ability to recover after the storm. So in the SAM panel, we scored in Ames in 2020. We did not score this trait per se, but I think there's a way to kind of study this. That is to look into the G2F dataset. Since you have this lodging of the same genotypes over multiple environments, that would give you the opportunity to explore if the lodging of a certain genotype would vary across multiple environments. And I hope that could provide some insights into this question.
Interesting. So would you measure them more than once over time to make sure that, I mean, I'm just thinking about this idea of recovery, because recovery happens over time, and how you would measure that exactly.
I'm aware of some studies that measured lodging over multiple time points, but for our study here, we scored like over 30,000 plants. If you do that more than once, I would no.
That's great. Thank you very much. Actually, thank you to both Adina and Zihao for both of your seminars. As a reminder, we've done these series a few times. And for those of you that are here, if you haven't spoken in this seminar series and you want to, let me know. If you have a friend or a colleague or somebody that you work with that you know gives a really great seminar, let me know, because we're looking for people to put into more upcoming Plant Phenomics Fridays. And as we get a panel of people that we could use to roll through another month of these kinds of seminars, we'll put it together. So with that, I'll thank not only our two speakers from today, thank you for the excellent seminars, but also for all of the month of August, our 4 seminar speakers, the support that we have from USDA, NIFA, and NSF, but also to all of you that are here. Thank you so much for joining us for Plant Phenomics Fridays. It makes a big difference to have a group of people that's interested and wants to have a discussion after a seminar, because we all get to learn a little bit more. Thanks so much. I hope to see you and interact with you again really soon. Bye-bye.